My name is Dan Norton, Product Manager in the DNA Applications Group at Illumina, and I have the pleasure of introducing our speaker today, Dr. Jamuar. Dr. Jamuar is an attending at the Genetics Service at KK Women's and Children's Hospital in Singapore. He is also the co-founder of Global Gene Corporation and serves as the clinical lead of Precision Medicine Initiative and Childhood Undiagnosed Disease Program in Singapore. He completed his genetics fellowship at the Harvard Medical School Genetics Training Program and also worked as a postdoctoral fellow in the Christopher Walsh Lab at Harvard Medical School. He's actively involved in research. He's authored numerous peer-reviewed publications and is presented at both local and international meetings. His research areas include dysmorphology, neurodevelopmental genetics, and associated molecular and cytogenetic technologies. He was awarded the Young Investigator Award at the American Epilepsy Society for his work on targeted resequencing on patients with epileptic encephalopathy. Lastly, he is also the recipient of the 2015 Sing Health Outstanding Young Researcher Award. And with that, I would like to turn it over to Dr. Jamuar.
Thank you, Dan, for the very kind introduction. Good morning, everyone. Thanks for joining in. Thanks to Illumina for organizing this webinar. As mentioned, I'm Saumya Jamuar. I'm a clinical geneticist at KK Women's and Children's Hospital in Singapore. And today I have the pleasure to talk about my work on using deep targeted next generation sequencing to detect somatic mutations in patients with brain malformations. This work was carried out during my postdoctoral fellowship in Christopher Walsh's lab at Harvard Medical School. In terms of disclosure, I'm the co-founder of Global Gene Corp. It's a data-driven genomic solution company providing solutions across emerging markets. And the opinions expressed in this presentation are mine and not opinions of Illumina. With that, let's start with some definitions. So somatic mutations. A simple definition of somatic mutation is that it's a post-zygotic mutational event. And what that means is that although the individual starts as a single fertilized egg, a mutation happens subsequently, and that leaves the individual with two or more populations of cells with distinct genotypes. And that's illustrated in the figure below. Somatic mutations are well recognized in diseases such as cancer, where different somatic mutations at both the level of the gene as well as the chromosome exist within the same cancer tissue. Somatic mutations have also been described in some genetic disorders such as McCune-Albright syndrome, Sturge-Weber syndrome, Proteus syndrome, and more recently a work done by Anne Paduri from Chris's lab on hemimegalencephaly. What's important to note about these mutations is that these are what we call obligatory somatic mutations, because these mutations, when present in germline, are usually lethal and do not lead to a live birth. So whenever we see these mutations, we usually see them in a somatic setting, and hence they are referred to as obligatory somatic mutations. In contrast, there are some mutations which occur both at the same amino acid in the germline as well as in the somatic state. And when they do occur as a somatic mutation, they lead to a milder phenotype. So in this illustration, the individual has a germline mutation in a gene called LIS1, which is associated with lissencephaly, a neuronal migration disorder. LIS1 is important for migration of neurons. When the mutation occurs in the germline, all the neurons are affected, and hence the brain has a smooth appearance or lacks gyri. In contrast, when this mutation occurs as a post-zygotic event, there are some neurons that have the normal LIS1, whereas some neurons that have the mutated LIS1. This leads to the formation of a milder phenotype called double cortex, which can be seen here. There's a second layer of cortex that's visible in this individual's MRI. Clinically, these individuals do present with a milder phenotype as well. Another concept when we talk about somatic mutation is timing of the mutation. And just going back on a very simple schematic of embryology of the human fetus, uh, we all start as a single cell and then subsequently divide into different components, the chorionic villus, the amniotic fluid, as well as the fetus. And around day 15 is when the germ cell layers are formed into the three germ cell layers, the mesoderm, endoderm, and ectoderm. And as we can recall from embryology, the, the brain comes from the ectoderm. The other tissues that come from the ectoderm include the skin and the buccal mucosa. The blood, on the other hand, comes from mesodermal tissue. This is important to note because a somatic mutation occurring beyond day 15 may only affect organs that develop from that germ layer. And that's an example in this mutation, which I refer to as a late somatic mutation, where the mutation has occurred in only the brain, in only a subset of neurons in the brain, leading to a right-sided hemimegalencephaly. And in this individual, if we test the blood sample or DNA from blood, we will not be able to detect the mutation. With that background, somatic mutations, we know that they exist. The question is, how common are they? And if they are common, how do we detect them? So just reviewing the techniques available for detecting mosaic variants. The first one and the most commonly used is Sanger sequencing. It's a technology that's been around for decades, but we also know that it has its limitations. And the limitations are that mutations below a threshold of 15 to 20% are either missed or not easily picked on a simple Sanger sequencing. More recently, whole exome sequencing has been used to detect these mosaic variants. Unfortunately, the problem with whole exome sequencing is the cost of getting adequate depth. So the alternatives are to use a paired tissue sample, such as in cancer samples, or if it's a single unpaired sample, then use liberal ion filtering during bioinformatics. The last technique is, and what I'm going to be talking about, is targeted sequencing. And some work done prior to my work showed that targeted sequencing was good enough to detect mutations at level as low as 1%, but that depended on depth of coverage. So one of the first few papers, a beautiful paper by Izawa et al., published in 2012, where they looked at three-point mutations in the gene NLRP3, and they diluted this mutation at different concentrations. And what they found was that at 1% dilution, at a read depth of 500, they were able to detect the mutant allele with statistical significance. So this was one of the first papers to show the ability of targeted next generation sequencing in a very limited fashion to detect somatic mutation at level as low as 1%. This was then followed by work from Bill Dobbins' group, where they looked at de novo germline as well as post-zygotic mutations in genes in the mTOR pathway, AKT3, PIK3R2, and PIK3CA. And again, they demonstrated presence of somatic mutations in these individuals. Again, they were able to detect mutations less than 10%. Some of them were even 1%. What was interesting was that when they looked at the distribution of mutation, somatic mutation within different tissue types, in this individual, the blood sample had only 12% somatic mutant allele, whereas the buccal swab had 41%. So clearly showing that certain tissue tolerate somatic mutations better, plus again going back to embryology, maybe because the buccal mucosa is an ectodermal tissue, it's closely related to the brain tissue. Lastly, what they required massive depths. These samples were sequenced at 200,000x. So what they did was they looked at a small set of mutations at very deep, very ultra deep sequence, and performed ultra deep sequencing to detect these mosaic mutations. So now that we know that there are somatic mutations in brain malformations, we know that targeted next generation sequencing can detect these mutations. The questions that I started off with was, how common are these somatic mutations in neurodevelopmental disease or disorders of brain malformations? And what is an optimal technique to detect somatic mutation, but in a larger panel of genes where we don't know what the mutation is and we are hunting for those mutations? So the patients were recruited from brain malformation clinic. This is a clinic that was started by Chris in the early 2000s and had patients since then, many of them who remained undiagnosed despite having a very defined phenotype on MRI. These patients were either referred from our local collaborators within the hospital in the Boston area, within US, and some even internationally. We excluded patients with known mutations. And more importantly, a point to note is that we performed this assay on leukocyte-derived DNA or DNA from blood sample. Unlike cancer, we didn't have access to brain tissue, even though we knew that there could be somatic mutations. We didn't have access to brain tissue unless these individuals underwent surgery for refractory epilepsy. So for most of the individuals, we were left with just DNA from the blood. And that was a concern when we started the experiment. In terms of the patient distribution, there were 4 defined phenotypes that we looked at, and the phenotypes were defined by the appearance on the MRI. The first image on the left is periventricular nodule heterotopia, and what you see is nodules along the ventricles. This is classically caused by mutations in gene filamin A. The next phenotype is a pachygyria phenotype, where there's absence of gyri formation and it's a smooth brain appearance, classically caused by mutations in NIS1 gene, double cortin, dyneins, and tubulins. The third phenotype was the double cortex phenotype, where you see a normal cortex and a second layer of an abnormal cortex, typically caused by double cortin. And the last one was the phenotype of polymicrogyra with megalencephaly, caused by mutations in genes in the mTOR pathway, as demonstrated in the earlier paper. With that, we had a total of 158 patients. We then used TrueSeq custom amplicon kit to design 2 panels. The first one, when we first started the experiment, was a much smaller panel, 14 genes, 600 amplicons, target region of 84 kilobases. There were 10 known genes and 4 candidate genes. 10 known genes, meaning genes where somatic mutations had been described in association with brain malformations, or where they were known to cause brain malformation in a germline setting, but never been reported to cause brain malformations in a mosaic setting. The second panel, we expanded it to 54 genes, close to 1,500 amplicons, and the target region was about 237 kilobases. There were 18 known and 36 candidate genes, and these candidate genes were identified through protein-protein interaction from the animal work that was being carried out in Chris's lab. The catcher regions included the exons, the intron-exon boundaries, and 10 base pair flanking sequences to capture the splice variants. We performed sequencing on a MiSeq. Samples were multiplexed to run on a single MiSeq flow cell. And again, this was more about trying to get adequate coverage to run as many samples. And based on our calculations, an average coverage of 200x was something that would allow us to confidently call a somatic variant. And so that's the target we chose. As a control, we had 5 germline controls. one negative control, and we also had some spike control where we used patients with germline mutations, diluted it with individuals without a mutation in that gene at levels of 50%, 10%, 1%, and 0.1%. Variant annotation was done using the MySeq reporter version 2.1.43, which had the somatic mutation caller function. And we arbitrarily defined variants as either candidate germline if the read frequency was more than 40%, or candidate somatic if the read frequency was less than equal to 40%. We performed Sanger validation in all rare protein altering variants. These included non-synonymous, nonsense, indel, frameshift, and splicing mutations. For germline variants, we performed segregation analysis to establish pathogenicity. For mosaic variants, we performed subcloning followed by Sanger sequencing of these colonies to establish the presence of the variant. In defining pathogenicity, these variants we required them to be either absent or very rare from the control population, including DB-SNP, 1,000 genomes, EVS, and at that point we didn't have exact, but now that would be another criteria. These variants had to be protein altering, so non-synonymous, nonsense, splice site, frameshift, and indel. And in silico prediction software predicted them to adversely affect protein function. So we use SIFT and polyfen2. In addition, for germline variants, we expected them to have to segregate with the disease. So either they arose de novo, the parents were unaffected, they segregated with the disorder if there were other affected individuals in the family, or inherited from a parent who had somatic mosaicism, or adhered to a dominant X-linked mode of inheritance where applicable. So with that, we went on and did an analysis. The first thing in terms of quality control metrics, we generated on an average 5 gigabases of raw sequence data per run. 94% of this mapped to the targeted region. The per base quality score was excellent for the first 100 base pairs and quickly dropped off after that. But the per sequence quality score was an average of a Phred score of 37, which was fairly good for what we were looking for. We then looked at our calibration samples, and this is one of the variants in double cortin gene, DCX. It was a germline variant, and when present at 50%, the allele fraction was 46%. At a dilution of 1:11, we observed allele fraction of 13%. At a dilution of 1 in 100 or 1%, we saw an allele fraction of 2%. And that was what our theoretical limit was, because when we looked at a dilution of 1 in 1,000, we were not able to confidently detect the variant. We then looked at our second calibration sample, which was a heterozygous variant in filament A. And again, we observed the same phenomena where we were able to confidently detect the variant at allele fraction as low as 1%, but not at 0.1%. In terms of coverage, 90% of the bases were covered at 100x and about 85% were covered at 200x. So on average, the depth across the samples was 300x. In our combined panel, we detected an average of 368 variants. But after applying filtering, only 57 rare protein altering variants were detected in 50 patients. 35 of them were at allele read frequency of more than 40%, and hence called as germline. 22 variants were below 40%, 5 were between 40 and 30%, and 17 were at allele read frequency at less than 30%. We then went on to do validation, and as expected, all the candidate germline variants did validate. We then looked at pathogenicity, and only 19 of them were truly pathogenic. One was a variant of uncertain significance. We then looked at variant at alternate allele read frequency of 30 to 40%. out of the 5, only 2 validated. 1 was pathogenic, and when we performed subcloning, we found that the variant was a mosaic, present at allele read frequency of 35%, and then we had 1 variant of uncertainty. Lastly, the interesting part is when we looked at variants which were at very low allele frequency of less than 30%, only 4 of them validated on Sanger sequencing. 2 of them were pathogenic, but the remaining 13 did not show up on Sanger validation. So we then went on to do subcloning and sequencing individual colonies, and we were able to successfully validate 5 of the remaining 13 variants. So a summary of the mosaic mutations in this table. We had 8 individuals who had a mosaic mutation, and this table is listed in increasing order frequency of the alternate allele read frequency. And as you can see, we could detect mosaicism as low as 5%. These mutations were either had been reported previously or were loss of function mutations and consistent with the mechanism of disease, and referred to as pathogenic. Overall, in 158 samples, we were able to diagnose 27 of them. So that gave us a diagnostic yield of 17%. What was interesting was that 8 out of the 27 were actually mosaic mutations. So about 30% of our study cohort actually had a mosaic mutation. What's even more interesting is that 5 out of the 8 mutations were actually missed by Sanger sequencing. And true enough, some of these individuals had even undergone clinical testing for genes, and they were told to be normal. But with next generation sequencing, we were able to identify these mutations and confidently elucidate the molecular etiology of their medical condition. One of these individuals had even undergone whole exome sequencing, and the mutation was missed. When we went back and looked at the data, the mutation was there, but it was the read depth was very low. It was only 4 reads that covered that region, and hence was filtered out during the analysis. So that again shows the advantage of using a targeted panel where you know where to look for the mutation. We then looked at the depth of coverage and whether that correlated with our ability to validate or not validate the variant. And what we found was that at a depth of above 100x, the variants did validate, and at a depth below 60x, the variants did not validate for somatic variants. For germline variants below 20x did not validate, and variants above 20x did validate. We then said, we have got alternate allele read frequency from 3 different independent technologies: next generation sequencing, subcloning, and Sanger sequencing. Is there a correlation, and would that correlate to the disease severity? And what we saw was that next generation sequencing and subcloning were closely correlated. So you can see the 2 graphs in red and blue having a high correlation. But when we looked at Sanger sequencing missed 4 out of the 8 variants, and in some of the variants, such as in this case, it was only on retrospective analysis that we found that the variant was there. It's because we knew it, but if we had just gone to look for that variant, it would have been easily missed. What was also interesting is that this variant here was actually called as germline when we looked at the Sanger traces. So the peaks of the 2 alleles were equal. Whereas when we did next generation sequencing and subcloning, the allele frequency was 35%, alternate allele read frequency was 35%. So again, this graph what it demonstrates is that Sanger sequencing has limitations when it comes to mosaic mutations, both when it's at low levels as well as at higher levels. This is an example of just a comparison of how relatively how much easier it was with the targeted next generation sequencing, because in this slide where you can see these are the next generation sequencing reads. This is just a representative image. It doesn't show all the reads, but it was quite easy to pick out that one alternate allele amongst the normal. When you look at the bulk Sanger sequencing, this allele is not present. It can't be seen. But when we did subcloning, we identified the normal allele as well as the alternate allele, confirming that this variant was present. And this variant did cause a protein, alter the protein, and was damaging. Similarly, in the second figure on the right, the variant can be seen quite easily, not seen on bulk Sanger sequencing, but again picked up on subcloning. In this individual, this is the individual where the alternate allele read frequency was about 12%, and on hindsight, it can be seen that there is a small peak there, and some people may say that, oh, they would, it can be called, but on most routine specimens, this would have been called as a background noise and probably not called as a variant. This variant was easily picked up, and it was de novo, as demonstrated by absence of the variant in the mother as well as the father. The next question that we asked was, yes, we found variants, but what if these variants are just incidental findings? And how do we know that, yes, we've identified the variants, we have validated the variants, these variants are predicted to be pathogenic or have been reported previously in either a germline or a mosaic state? But how can we be sure that these variants are true and not just incidental findings? So what we did next was although we did not have a control group, what we did have was we had 4 different groups of individuals, which we could lump together into 3 major groups. One was the double cortex or pachygyria related genes. The next was the polymicrogyria related genes, and the last one was periventricular nodular heterotopia related genes. And what we showed was, we said, let's look at what's the frequency of finding a synonymous variant or a non-protein altering variant in these samples. And as you can see by the bar graph here, most of the across the different phenotypes, the distribution was fairly similar. We then said, okay, let's look at rare protein altering pathogenic variants. And what we saw was that the variants were very specific to the phenotype. So in this example, you can see that all individuals in this had a double cortex or a pachygyria phenotype, and the variant was in a double cortex related gene. Similarly here, these 2 individuals had a polymicrogyria phenotype, and again, the variants were only seen in polymicrogyria related genes. And lastly, in this scenario, the 8 individuals had variants in periventricular nodular heterotopia related genes. There was one individual here who had a variant in a PMG related gene, and this was inherited from the father, and we could clearly call it as a benign because father was unaffected. But overall, what this demonstrates is that these variants were fairly specific for the disorder that they cause, and not just randomly distributed across different phenotypes. The last thing we looked at and said was, great, now we have identified pathogenic mosaic variants. Is there a way can we correlate these variants with the phenotype? And what is demonstrated here on the top is individuals with a variant in a mutation in double cortex gene, and in the bottom row, a variance in the lissencephaly gene. And as I mentioned earlier, both lissencephaly and double cortin cause a neuronal migration disorder. And depending on the proportion of neurons with the mutation, the double cortex will depend on the number of neurons that have the mutation. So in the first example here, where the allele frequency is 5%, there's a very thin band of double cortex compared to this individual who has 15% allele frequency, and the band, as you can see, is much thicker compared to this individual where the allele frequency is 50%. And this is an individual, double cortin, as some of you may know, is an X-linked gene. So in a male, in a germline state, it's 100% of the cells that have the mutant allele, and this individual has a neuronal migration disorder that affects all neurons and has a pachygyria phenotype. So what this demonstrates is a fairly nice correlation between the level of alternate allele frequency in the blood sample with the severity of the double cortex on brain imaging. Similarly, in the bottom row, again, it can be seen that the individual with 16% allele frequency has a much thinner double cortex compared to someone who has 26% and someone who has 50% allele frequency. So there was a nice genotype-phenotype correlation as well in the proportion of cells in the blood with the alternate allele and the brain imaging. So just to end the talk, I'd just like to conclude that we successfully used targeted next-generation sequencing in individuals with brain malformations with an undefined molecular etiology, and we were able to get a diagnosis in 17% of them. This diagnosis ranged from 10 to 30% across the 4 phenotypes. We identified germline variants in 12% of them, but we identified mosaic variants in 5% of them. More importantly is that 63% of these mosaic variants would have been missed on Sanger sequencing, and one was even, in fact, missed on whole exome sequencing. And the ability to detect mosaic variants is as low as 5% in individuals with brain malformation. That I'd like to thank you for listening, as well as thank Chris for giving me the opportunity to do this work, and people in his lab, as well as the various collaborators who worked on these samples, either contributed or worked in this project.
The first question for you, Dr. Jamuar, is coming from the audience is, in my research, I have similar issues with Sanger sequencing with respect to limit of detection. It looks like most of the value of your study was generated through targeted resequencing. What was the primary reason for the use of Sanger sequencing in this study? Why not just use targeted resequencing?
That's an excellent question. And so for our group, we decided to use Sanger sequencing because that was the gold standard used previously. Some groups have actually used an alternate platform. The main reason why we didn't just use targeted resequencing again was because we know that machines have their inherent biases and errors, and it's important to be cognizant of that. And if while designing an experiment, one wants to use next generation sequencing to validate these mosaic variants, it's important to use an independent platform. And so that's why some groups have actually used ion torrents machine to validate these variants. But for our study, because we were more familiar with cloning, and it is much simpler protocol than designing a new assay on a completely different machine, which again we didn't have access to. We decided to go with Sanger sequencing and cloning.
Will resampling of different regions cause the discrepancy?
I'm not sure I get the question. So resampling of the, because these were all from blood, so I wouldn't expect any differences if we took another blood draw and repeated this experiment. In terms of sampling from different tissues from the same individuals, yes, we did see a difference in the proportion of alternate allele reads across different tissues, and the one that correlated that was most distinct. So saliva and blood had similar amounts of alternate allele read frequency, whereas the buccal swab was significantly higher in individuals where we did have access to those samples. In one individual, which I mentioned in the introduction, was the individual with hemimegalencephaly. We had the opportunity to sequence the brain sample as well as the blood sample, and we found the same level of proportion of variant through next generation sequencing that we had detected using single cell sequencing. So there wasn't a discrepancy when we did, in two different experiments. So the proportion was the same in the same sample. I don't know whether that answers the question.
Okay. You stated that in your results that you did not functionally validate all germline and somatic mutations and that your criteria for pathogenicity was conservative. If you were to redo the study, would you make your criteria for pathogenicity less stringent, and what would that accomplish?
So, so that was one of the limitations of this study, that because we were doing a screening sort of experiment where we were trying to define the question not so much about finding new alleles, finding new genes. The question was more about, can we detect somatic mutations and at what level, and what is the burden of disease? So when we designed the experiment, we did not go into functionally validating any new allele. Again, if we did, or if we used other less stringent criteria, our yield would be higher. But we decided to be conservative because that was not the main aim. And the aim was more to just demonstrate that somatic mutations do occur at a significant frequency that is clinically relevant to the patients that we see in the clinic. So yes, definitely, if we make the criteria less stringent, our yield will be higher.
With NGS, when you see different mutations, do you apply a threshold of quality to weigh that different call?
Yes, so as I mentioned, we had about 358 variants per sample, and we did apply a quality threshold to filter out some of these variants. Most of these variants were recurrent variants in regions which had areas which were homologous, which were not mapping correctly, and over time we were able to recognize these common recurring variants. But so yes, we did use a quality threshold to filter out some of the variants. But all the variants but it was fairly liberal filtering criteria. It was a pass score. It was a variant that was not clustered within a region. So if within a window of 10 base pairs, if there's 3 variants, then that variant was excluded. Yeah. So we did use quality score, but all the variants here were of good quality. had good quality scores.
Has there been any further study of the mosaicism in cortical malformations?
Yes, so not so much in cortical malformation. So one of the projects that I initiated and then was taken up by one of the grad students in Chris's lab, Alyssa Digama. She published her work in Neuron in December 2015, where we did targeted resequencing in individuals with autism, and again we did identify regions. We did identify mosaic variants in individuals with autism. And in that experiment, we were actually able to have access to brain tissue from individuals with autism who had passed on for some unrelated cause, uh, and we were able to identify these somatic variants. So that's one that's been done. The second one that was just uh, I did not attend ASIG this year, but from Brian Roke's lab, they looked at mosaic variants, and what they found was that there was an excess of synonymous mosaic variants in individuals with autism. So although autism is not strictly cortical malformations, but in a neurodevelopmental phenotype, there have been studies that have looked at that have attributed somatic mutations leading to disease. The last paper is by Ellison et al. That was published in Nature a couple of years back, where they looked at individuals with intellectual disability, and again, about a third of their uh, families had a somatic variant. And in that paper, they actually used ion torrent to validate those variants.
What would be another way to test that these mutations are related to pathology beyond the distribution test between synonymous and non-synonymous variants? As you mentioned, preliminary findings show that the synonymous variants may contribute to disease, whether in germline mutations or somatic mutations.
Yeah, that's an excellent question, and and just going back to the earlier question on functional validation, uh, definitely with the ability to do with CRISPR and gene editing, it's possible to create an animal model where you could induce a somatic mutation and replicate the phenotype. And that would be a more definite experiment in terms of confirming the pathology of the variant, uh, of a particular variant to the disease. But again, as I mentioned, we hadn't designed the study for that purpose, and we we used some statistical methods to ascribe pathogenicity to those variants.
Have you looked at any other techniques for low-level mutations besides NGS, such as digital droplet PCR or cold PCR?
Yes. So again, so Chris's lab is very actively looking at different technologies of detecting somatic mutations, and a paper that came out around the same time as my paper in Cell Reports looked at single-cell whole genome sequencing to identify somatic mutations, somatic copy number variants in brain tissue. And what they found was that there were individuals with somatic copy number variants, not to the level of aneuploidy that had been reported previously by experiments using fish, but with single-cell whole genome sequencing, there were individuals with multiple somatic copy number variants. And one of the individuals even had a somatic variant in the Prader-Willi region. Uh, so, so this is an area where uh, different techniques are being developed. ddPCR is another technique that we have looked at, and with some success. So yeah, so yes, we have looked at other techniques, and we have been successful. And I think there is There is there is some interest in this area, and I think there'll be more and more refinement of techniques and new techniques coming out looking at somatic mutation.
If you sequenced brain tissue, would the threshold for germline versus somatic mutation be different than 40%, and why?
So in the autism experiment where we had the brain tissue, we still kept the threshold at 40%, because we know that there is an inherent bias in these capture-based experiments, where the reference allele tends to be represented more than the alternate allele. The threshold also depends on the depth. So if someone was sequencing to the depth of 5,000x or more than that, then definitely the threshold will change, because your confidence interval for calling a variant germline versus somatic would differ as well. So definitely if the depth was different, and if you see that, in my mind, the depth is a more critical component in determining the threshold for calling something germline versus somatic. And when we looked at we did a simple chi-square test to determine, and we realized 40% was the threshold, where at the depth that we were achieving, we would be able to call mutations germline versus somatic.
So a special thank you to you, Dr. Jamuar, for taking your time to talk to us today. That was very interesting work, and we're very happy to have you join us and present your work.